Hello, my name is Antoinette Cordova and I'm with Emit Imaging. And today I'll be talking about cryofluorescence tomography or CFT in neuroscience. So you might ask, what is CFT? CFT is an imaging technique that captures 2D fluorescent and white light images of ex vivo serial sections and compiles them into 3D images. As can be seen in the cynomolgus head, a whole animal model, a mouse, and a high resolution image of a mouse kidney. So how do we accomplish this at Emit Imaging? We do so with our cryofluorescent tomography platform, Xerra. Xerra features a 12 megapixel high resolution camera, a 10 micron slicer, 6 high powered lasers ranging from 470 to 780, and 7 narrow band emission filters. This allows for our platform to offer 3D high resolution imaging that is quantitative, features micro resolution, automation, and ease of use. So how does Xerra integrate into your workflow? Xerra does so in 5 easy steps. First, a whole tissue is embedded into a block of OCT. The block is then mounted into the system and imaging is started. A camera images the block in fluorescence and white light, and a tissue is then sliced and imaged again, and this process is repeated hundreds of times. Lastly, the image stack is error corrected and reconstructed into a 3D model and analyzed. So in 24 hours, you get an exponential amount of data that gives you insight, for example, into the comprehensive biodistribution of a drug or a reporter group. Tumor burden found in a mouse xenograft model, the multiplexing of lasers. Xerra truly features the maximizing of data from one animal with complete data sets. So let us now focus on the applications of our CFT platform, Xerra, and today we will be highlighting neuroscience. So as we look at neuroscience, we're going to be discussing pharmacokinetics in the brain. Here in this experiment, we were looking at the delivery of antisense oligonucleotides or ASOs that were labeled with a size 7 and evaluated in mouse brains. Here we see the time course study showing the biodistribution of the ASOs over time from 1 hour to 12 hours. A closer look at this study shows that researchers can really track the movement of dyes and molecules throughout the body, specifically in this example of the brain. Another great feature of the Xerra is that the fluorescent imaging is also co-registered with white light data that can be used with brain atlases to provide information about the anatomical distribution of a drug or reporter group and its subsequent analysis. Another great feature of the Xerra in our platform of CFT is that it complements the histopathology workflow. Here, we see that a section can be removed in the automated process by stopping the Xerra at an area of interest and collecting the sample with a specialized histology tape. The histology tape can then be placed on a glass slide and imaged. So ultimately, why choose the Xerra? The Xerra is a powerhouse, truly with the many features that it offers, from high resolution camera to 10 micron slicer to the many lasers and the emission filters. It's also very versatile. It supports the fluorescent imaging from 470 to 850 nanometers in a wide range of fluorophores. In addition, the Xerra is automatic. It automatically slices images and compiles these 3D images and data sets. And lastly, the Xerra is cost effective. It replaces many weeks of lab work, tissue processing, microtome slicing, and slide preparation in addition to consumables. So if you would like more information about the Xerra or CFT, please feel free to contact me or us at emitimaging.com. Thank you.